When I always put this world's most diverse thing up there, it is certainly true for the major crops. It's probably not, it's not true in the biggest scheme. Probably things like grape and a few other crops probably challenge it for being extremely diverse. Another problem is when I talk to vertebrate communities, I want to show you how diverse this species is. But compared to the microbial world, maize is, it has a whole microbial genome that's moving around, and it's part of that world. And it's extremely diverse in that regard. But let's talk a little bit about maize diversity and what we can do with that diversity in terms of breeding, and what we can use with that diversity to understand complex genetic architectures. In this realm, I've been collaborating with a great group of colleagues over the last decade to really get to some of these questions. And I just want to highlight that this has been a collaborative project among Jim Holland, Mike McMullen, Sherry Flint Garcia, Doreen Ware, Chi Sun, Steve Kresovich, and John Dobley. And we've gotten most of our support from the U.S. Department of Agriculture, ARS, and the NSF Plant Genome Program. So I'll go through, I'll tell you a little bit about maize diversity, and then where we've used next generation sequencing to move our knowledge of the genome into what we call HapMap version 2. And then I'll go into a little bit about past selection, domestication, and then how we'll understand a few lessons about what's gone on in the past in selection, and then where we can use today's technologies to really move selection forward, and we can really accelerate breeding. So you and I, as you probably know, only differ from one another in about 1 in 1,000 base pairs. And, chimpanzees are, say, 15-fold more divergent from us. But essentially, any two corn plants, and I'm just talking here really at the genic region, is as different from one another as you and I are from chimpanzees. And so if you start adding up the numbers for that, we're starting to talk about and I'll show you evidence that, we have at least 50, 60 million common variants. And there's got to be hundreds of thousands of functional variants mixed in there. And essentially, the opportunity we have is to make the connections between each of those functional variants and important agronomic traits, and also then use all that functional variation to breed. And, but it goes beyond that. And a couple key studies done in the last decade, first by Hugo Dünner and then Michele Morgante, their groups, they essentially were able to show that if you look at small regions in the genome, only about 50% of the genome is really shared between any two maize lines. The retroelements between there have moved around. They're not shared at all. I mean, if you were looking from a large scale, all you got to look at is the genome, you'd say, these aren't the same species at all. It isn't until you look at the morphology of the plant that you realize this is the same species. And so we know that a lot of this is going on, and not only are they moving retroelements around, they're certainly moving around gene copies, they're moving around enhancer elements, and all sorts of other low copy fractions of the genome. And in comparison, I always view human, and when they talk about copy number variants, as these very boring species that virtually nothing happens. And so that diversity, though, comes from essentially having large effective population sizes. Very similar to the microbial world, there's it's not quite the scale of microbial effective population sizes, but they're large. And maize diversity does go back a long time. Some of modern maize alleles, some of them probably diverged from one another about 5 million years ago. We know the sister genus diverged, say, about 3 million years ago, and there's shared alleles with this sister genus even. And most of the maize species started diverging back 1 to 2 million years ago. And so really maize domestication, which happened about 10,000 years ago, it's just a little blip in the history of maize domestication. It was certainly important. We lost some diversity during it. But many of the important functional alleles, and we also can mine essentially all of the alleles going throughout this time period, and they have evolved and adapted to environments over a multi-million year time period. And just this gives you a little feel relative to the human variation. This is why most human variation probably only dates back a couple 100,000 years. So it's a very short time depth compared to what's captured in maize. So we wanted to start quantifying this, and this grew out of a HapMap version 1 where we focused at the gene-rich fraction of the genome. And in this effort, led by Doreen Ware, Jeff Rosser-Barrow, and then a couple colleagues at the Beijing Genome Institute and CIMMYT, and I would want to highlight that we got support from a number of different agencies, and Sean Kepler, for example, was able to bring some very important sequence from JGI for a couple of the lines that we'll be talking about here. And in the analysis I'll be talking about in this first section, there was a very talented postdoc with Doreen Ware's group, Jeremy Chia, who has, although 40 people probably participated in the analysis, he was the one who did the most and some of the best work on this project. So what is HapMap version 2? Essentially, we want to really increase the breadth of the samples we're working with, and we're working with teosinte, which is maize's wild relative, land races. So these are the varieties that were growing, essentially dominated all maize agriculture until about 50 or 60 years ago, and then improved lines, which are the ones that are inbred that breeders use to make hybrids. And very key that John Dobley made for us inbred versions of teosinte and land races. So we were able to phase the chromosomes for these normally outcrossed varieties. Maize is normally 100% outcrossing or nearly 100% outcrossing. It's only through manipulation that we can self them down. And we have inbred versions of teosinte and land races. We did paired end Illumina sequencing, generated lots of sequence, and had about 5x coverage over each of these lines. And remember, we only have they're all inbred, so there's only effectively one allele at each one of these. So if you're in comparison to other systems, it's would be kind of like having 10x. The first thing I want to highlight is it's a warning about this, and one of the really nice things we were able to do is we were able to take the exact same data set, and we ran it through Cold Spring Harbor's SNP calling pipeline, and we ran it through Beijing Genome Institute's SNP calling pipeline. And the key thing was only 50% of the sites agreed where they called SNPs. And we were talking the exact same QSeq files were handed off into these things, and they only called SNPs at the same location 50% of the time. Now, when they did call them, they certainly agreed in the SNP calls and everything, but each of them are tuned to various alignment tuning parameters, and one had probably a little bit better quality control filter. The other one was more sensitive to different homologies and everything, but they don't agree very much. And so when we ran into that, we said, we've got to be very careful on how we're calling SNPs here. And so what we did is we called about 160 million SNPs, and they really do, and I really do think most of these 160 million, and even many of the ones that don't agree, really do exist somewhere in the genome. This is not a sequencing quality problem. This is a problem of where you take that sequence and where you put it on the genome. So I think most of it does exist. And so what we did is we used, similar to what Ruth was talking about, we used regions of identity by descent and a number of population and genetic attributes such as linkage disequilibrium and heterozygosity, since we have inbred lines, to set up a series of filters that allowed us to define which polymorphisms were likely mapping to the right regions of the genome and which ones were likely do exist, but they're someplace else in the genome. And we don't know where, but they're someplace else. And once we did that, we came up with a very useful and very accurate quality control. We can't eliminate all the errors, but now we have a very good quality control parameter that correlates very well with IVD regions. And so through that, we were able to keep and collect 56 million SNPs that seem to be very useful. So there's more out there, but we can right now work with about 56 million. The first surprise that came out was when we sequenced this outgroup, the sister genus, to maize. And our prior knowledge is relatively limited. We knew it had a bigger genome. We knew it had 8 more chromosomes than maize did. One of the things that's always excited those in germplasm diversity is that this species is adapted to environments all the way from northern Iowa all the way down to South America. So its natural adaptations is far wider than maize's wild relatives, which mostly grow just in central Mexico. So if we were able to use this diversity, it would be incredible for maize improvement. And it's perennial. It's sometimes apomictic, which means it can make seed without sex. And it was often considered an allopolyploid. You know, they said, well, it's got 8 more chromosomes. That came from some other species, which happens all the time in plants. So we sequenced it. Nope. Trypsicum's genome is pretty much like Zea's genome. There's no chromosomal duplications. There's almost no duplications anywhere across the genome. There's almost no difference in the gene content between the two species. And so that's suggesting, despite all these different adaptations, the same, essentially, genetic architecture is at play here, which also means we can then manipulate that genetic architecture or even pick up the versions of the genes that are in Trypsicum, stick them in maize, and we should be able to enlarge the adaptations we have. Now, the retrotransposons between the genes have entirely changed. You know, they've all swapped out, and they're all doing different things. And so those are totally different. There's even shared nucleotide variation in genes, and somehow the chromosomes just seem to have split apart. That's essentially now our interpretation of what happened here. But this now opens up a whole new broad set of germplasm, and essentially it was kind of relegated in the past until we did this genomic analysis that said, well, there's not much we can do with this thing. But now I think it's opening up people's eyes to say, hey, we can do something with this sister genus. I previously mentioned that there had been some very nice work looking at structural variants in maize, and just recently another group did an array-based analysis to look at how much there was in maize, and they were seeing copy number and presence-absence variation at, say, 10% of the genome. And so we tried to use our read depth information on where we were seeing lots of reads in various regions of the genome versus not. And we were able to compare it to some of the prior work, and it seemed to agree fairly well. And so what Jerming then did is he set up a contrast using a high-depth library, a 20x library as a reference, calculated read depth ratios across the genome in 10 kb windows. And as you can see, this is against one of our favorite maize lines, and there's massive amounts of read depth variation all across the genome. And when we quantify that, essentially 97% of all these 10 kb windows across the genome have at least 2-fold differences in one sample or more in read depth variation. Now, some of those could be caused by regions elsewhere in the genome. So we then took those same read depth ones and said, how many of those map locally? Essentially, can we find a segregating SNP that we know does exist there? Does it correlate with that read depth variation in that local region? And in 80% of the cases, we can confirm that that read depth variation is segregating and is locally associated. So essentially, our take home from this is the whole genome is in flux in terms of how many copies of the gene you have, whether you have the gene or the regulatory element. The genes are more conserved, but every other space of the genome is definitely in flux. So given that contrast, we then wanted to move into seeing what happened in terms of selection in the last 10,000 years. And so we did a domestication analysis, and we focused in on half the SNPs that are well represented across all these individuals. And I want to highlight that Matt Hufford and Jeff Rossabaro's group led this analysis, and he's done a beautiful job in doing this work. And the first thing we've done, and we've done similar types of analysis like this over the last decade or so, is to ask the question, how much diversity is in the wild relative, Parviglumis, relative to the land races, and then relative to improved lines? We haven't really had good inbred land races until very recently, so we weren't able to make this last contrast very well. But we saw the normal drop in diversity that we do from maize's wild relative down to maize, about a 17% reduction in diversity. But the kind of surprising thing was that we actually saw a very little drop in diversity in SNPs from these land races that are distributed across North and South America and improved lines. And I do want to highlight, the improved lines we're talking about here are not U.S. elite material. These are improved lines that include those, but also include varieties that have been developed in Thailand, throughout Mexico, and other regions. So they're much more diverse than what's grown just in the U.S. But it's suggesting that those breeders who've been trying to capture diversity out of land races have been actually quite successful. There's now lots of good, useful variations sitting in our land, in these improved lines, these diverse improved lines, that we can really start to harness and use for breeding. So I think that's a very exciting, a second exciting result that we get out of this. The breeders have done a good job so far. They didn't know what they captured, perhaps up until now, but they did a good job capturing that diversity. Then we can look at, do a genome-wide scan, looking for selection targets. Up until now, we had some ideas of how many regions have been targets, and of course, some very key domestication genes have been identified, but we didn't know exactly which ones they were. So Matt did a scan across the genome, and he used this XP-CLR approach to detect selection. And it's compared to fairly robust to bottlenecks, and it's able to determine some selection sweeps that are based on standing variation. In general, this is one of those plots of selection going across the genome. There's lots of them. The take home is there's about 500 regions, 500 domestication targets we can pick out from this. And then I'll give you a couple examples here of what those look like. For example, one of the domestication targets that we've seen, and it's been very nice to be able to contrast what we're seeing in maize versus rice to see if things are kind of tuned well. And for example, we're finding a couple genes in the flowering time pathways that have been definitely targets of selection, including one that had also been highlighted in rice, but we hadn't picked up so far. But yeah, as you can see, there's a big difference, and it confirms that we've seen in maize a big increase in expression relative to maize's wild relative teosinte. So we've also looked for improvement genes. One of the interesting things is that in the pathway involving gibberellins, this is the pathway that was very important in the green revolution in rice and wheat. It turned out in the improvement loci that we found, essentially the ones between the land races and improved lines, that a number of genes have been targets of selection. And so the gibberellin pathway in the last few centuries has definitely been very important in the improvement of maize. And we've got a number of genes we've now identified have been targets during that. So in general, it looks like these whole genome scans, we're getting reasonable genes out. And so now the opportunity is, some of these genes, you know, they have no diversity left, and that may be good, maybe that's bad. In some cases, we probably want to now go back to that wild relative and maybe look for more genetic diversity. And these also would be excellent candidates for taking, making into transgenes and up expressing them and down expressing them, because they don't have very much diversity left in maize. And so you either need to integrate some more from the wild relatives, or you need to make transgenes for these. But now there's 500 good targets. It's time to start manipulating those. Okay, but there's still a lot of variation left in maize. And so we were starting to ask that question, and Jerry Tuscan was kind of mentioning this: what regions of the genome play an important role for controlling quantitative variation? In the last year, a number of studies have been working on leaf architecture, disease traits, have done some very nice job doing genome-wide association analyses to identify key genes in some of these pathways. But we only were really working with a set of SNPs that were mostly focused in the gene-rich fraction of the genome. And so as we've expanded our survey now to include the whole, a wider swath of the genome, nearly the whole genome, what genes are playing an important role there. First thing I want to highlight is a little bit about how we do this. We do association mapping a little bit differently. We do the regular type of association mapping where you take a series of unrelated individuals and you do it that way. But the other tool that we have is what we call the Maize Nested Association Mapping Panel. And what this is a series of 25 diverse inbred lines, which we now know actually capture a good swath of the diversity. We've crossed them, though, to one reference line That was very important in U.S. maize breeding. And then we created 5,000 inbred lines from these. And essentially that allows us to break a population structure, create a common reference, agronomically allow us to put all these plants in the same field, which we couldn't do before. And then with lots of colleagues working together, we've been able to grow millions of plants and look and evaluate millions of plants. And essentially together, through this whole community's effort, we've been able to create essentially the largest genetic dissection system really ever put together. And it's given us some unprecedented power to identify some very small effects. And we need that because it looks like maize diversity is made up of lots of genes with small effects. And I'll give you a couple examples here. But for example, maize flowering time. We take two inbred lines, grow them in the same environment. They can differ by 2 months in flowering time. But when we look at what genes control them and how many genes control them, at least 50 genes are controlling this, and each of them seem to have small effects. We're talking on the order of hours. The allelic effects are often less than a day in terms of what their additive effects are. And so it's just by stacking up lots of gene and loci with minor effects that we get this tremendous variation. And we see that, for example, I, for example, might have thought that upper leaf angle, the angle at which a leaf is held up in would be much simpler. No, it's 30 or 40 genes at least, and each gene in each variant is only controlling a couple degrees. And essentially, you can see that this natural variation is there, and it allows adaptation and selection to work to exquisitely adapt a species to its local environment. You don't have to make jumps of 50 degrees in leaf angle. I'm sure that, and occasionally that may be adaptive, but in general, over millions of years, you want a more finely tuned system. And I think that's what's kind of evolved, is a system that is much more finely tuned. So what regions of the genome seem to be important in controlling these? What we're finding is we can then kind of break up our SNPs into whether or not they're coming from HapMap2, which is this whole genome-wide scan, HapMap1, which is the ones that are more gene-focused, and the read depth variants. And the first picture we see for a series of different traits is that, yes, it was really good that we did HapMap2. It explains a lot of the SNPs now from GWAS are coming from that. But the main reason is that they're providing information on a much wider swath of the genome. Many of the HapMap2 SNPs actually did a pretty good job, and if they were removed, they would explain a lot more of it. But we're finding better variants now with this HapMap version 2 SNPs than we were before. But if you look at the relative importance if you were to break out the SNPs by their relative importance, then we get a very different picture. Essentially, the gene-rich fractions SNPs, these are HapMap version 1s, they are enriched about 4-fold over their present, what we would expect by random chance to be in the model. And then if we look at read depth variants, okay, these are these copy number variants across the genome, they vary from they're enriched anywhere from 4 to 18-fold. So that when you gain and lose a region of the genome, it does matter. It isn't causing the plant to have giant QTLs at each one of those, but it's causing a lot of these small effects are coming from probably enhancer elements, copy number variants across the genome, and so they do matter. And I suspect as we get better ways of quantifying and copying these read depth variants, those numbers would actually just move up in their importance. So that's where I'd love to have PacBio data, things like that, so we can really understand where all these read depth variants are and be able to break them out accurately, because I think we're only starting to see the tip of that. So the question now is, we have all this variation. What can genomics really do to accelerate this breeding? So we're already in this process, but this is actually the evaluating the natural variation in the long run is one of the really expensive aspects of it, because you got to grow millions of plants out in the field. Then we need to create mathematical models to map the genotype to phenotype, and I think we're actually making some pretty good progress in this area, and many people in the breeding community have been working on these approaches. I'll show you in a second. Then can we predict phenotype? And then if we can predict phenotype, then we can move breeding very quickly, and I'll walk through some of those steps. The first thing is we can predict. You might think with all these small effects, you can't predict anything, but that's wrong. Actually, those small effects are actually quite additive. If we add up all of our predicted QTLs and we say, when would one of the parental lines flower, we get very accurate predictions. And essentially, we're often at levels that are pretty close to our total broad sense heritabilities. And it doesn't really matter what trait we've done. We've also done disease traits. For some of the components of these component traits, yield is harder, but for the component traits of some of these adaptations, we can very accurately make predictions. So if we're really going to then take this out into the field and start really accelerating things, what we need to do is be able to identify the right crosses to make. There's hundreds of thousands of maize varieties, and some of the key things is, what do you cross? You can't do everything. We need to make genotyping cheaper than it is to grow a plant out in the field, and I'll show you how we're doing that. And then once we have those two elements in place, it really then becomes an engineering process of figuring out what segments of the genome we then put together to get the varieties we want. And when you do maize breeding, there's thousands of environments you're breeding for. It's engineering for thousands of different microenvironments across the globe. And so it then becomes that matching process. So one of the key elements we needed was really low-cost genotyping that wasn't skewed by prior knowledge of diversity. We've got this incredibly diverse species. If you devise your genotyping approach based on essentially sequencing of two lines, it doesn't really tell you anything about the true diversity out there. So we wanted to use it, drive it out of genotyping by sequencing. This is an approach similar to lots of people who are doing bar-coded sequencing approaches, and I think the main thing we've done is make them more efficient. And what we do, the protocol is online if you want to read more about it along with the paper. But essentially, once we plate out the DNA, we put the adapters. We do everything essentially in a single well, very similar to what AFLP does, and it's very straightforward. We throw out lots of steps that people are worried about, size fractionation, things like that, because they don't really seem to matter for high throughput genotyping. We use variable length barcodes to get through some sequencing quality issues. And essentially, once we do that, we can pull the DNA, we can clean up the DNA. If it looks good on a run, then we know to go ahead and sequence it. So essentially, we've genotyped about 8,000 lines of maize this way. About 14% of the tags are probably too repetitive to do much with. But through this process, we've now identified about 8.8 million frequent tags, and we can genetically map over half of them. And about 1 million of them are telling us about SNPs that are segregating at the small portion of the genome that's always pretty much shared. And then there's millions of presence-absence variants that we're starting to identify. They're telling us about those copy number, read depth variants and everything. Right now, at 96-plex, we get back about 1 million tags per line. So, and if you look at this in terms of cost, we've now been able to move the cost well below what our field trial costs are. We're currently running 96-plex. You include the sequencing, including depreciation costs on the machines, labor, reagents for DNA preps, and so on. We're about $20, and we're shifting up to 384-plex, and then we'll be at about $10 a sample. And so at $10 a sample, there's some things you can do now that were just inconceivable 2 or 3 years ago. And so the first things we're doing, essentially we're genotyping every key genetic mapping and breeding population we can get our hands on, and working with colleagues across the world and getting the protocols out so that we're all kind of using the same basic system. We're working with a series of maize breeders for sub-Saharan Africa to genotype all their key maize and sorghum lines. And then the seed project at CIMMYT has picked up on this type of approach, and working also with Anjay Killian, who developed the DART approach. They're now shifting over to a very almost identical approach as what we're doing here. And the two groups are going to be working together to help CIMMYT essentially genotype about 400,000 public maize and wheat varieties that are essentially the major germplasm resources for breeding around the world. And But this number of 400,000, that's just kind of the beginning, because once we start applying breeding, we want to look through every year hundreds of thousands of varieties and throw out the bad ones. And so let me explain how that works and why this, just by genotyping, this is powerful. Essentially, this is the old cycle that people used to do breeding on. And it's the dominant way that breeding in maize occurred during the 20th century. And people would make and it was very successful. I mean, maize yield was gone up 8-fold in the 20th century, going through this cycle in the U.S. But you make crosses. You then inbreed those lines, and then you do small-scale trials to figure out what were the right lines and good lines. And then you make large-area trials to see which ones hold up in a number of environments. Those that worked would then get packaged up into making hybrids and be sold commercially. But if you look at this cycle, took on average, say, 5-7 years to do. And in terms of evolution, you're only making a gain every time you make a cross and select. So that cycle, it took 5 to 7 years to occur because it took them a long time to figure out what was good and what was bad. And now, in genomic selection, the cycle time changes. We can now make crosses and genotype them every 4 months. You can run trials and make crosses in Hawaii. You plant the seed. Every 4 months, there's a new set of seed, replant it. And so this basic engine of evolution now can tick at a rate that's more than 10 times faster than what we did during the 20th century. And the whole key, though, is whether or not we can make that mathematical model that's based on genotype and phenotype mapping good enough to predict which seeds to keep out of these crosses and go back into that advancement. And so just because you got a good line here and a good genotype doesn't mean 4 months later you're ready to go sell seed to farmer. It still needs to be taken and making an inbred. You want to definitely, your model isn't going to be correct all the time, but you just want to pick that top tail. You want to pick that top 1%, 5% of the ones that have a chance of being good. And then you still have to go through all of these steps, but that the central evolution cycle of evolution here, which is what breeding is really about, now has an opportunity to tick at 10 times the rate that it did before. Because of some inefficiency statistically in the model and getting ahead of the model, at least initially, people don't believe it can move much more than 3- or 4-fold. But if we had total knowledge, we could make that engine tick 10 times faster than what it does normally. And so I think we're at this extremely exciting point in breeding. And we can, in perennial biofuel crops, this cycle time, or tree crops, this cycle has gone like twice in loblolly pines in the 20th century. We can make each of these crops, especially the longer-lived ones, tick as fast as we can make crosses. And so, as long as we can build in breedability, I think we can make incredible gains. So I'm going to end with this last slide here, and I think it's really up to the, society and a discussion with geneticists as what should and can we do in the next several decades with these great new tools. Because as I was mentioning, the hard part is training the model. That's expensive. That model doesn't get trained for free. You don't grow millions of plants and look at phenotypes for no cost. And I think one of the goals, Monsanto has said it very clearly, and I think it's very doable. I think we can double yield in maize. And I suspect we can do a lot better in a number of other species that haven't been worked on as hard as maize has. In the developing world, I think we can go even beyond doubling it. I think we should be perennializing our crops in order to save on soils and make more useful, more use of our, keep nitrogen, get access to some of the deeper phosphates that are down there. We can really do a much better job of breeding crops to cope with drought and use nitrogen efficiently. And I think we also have a great opportunity to biofortify our crops for improving nutrition around the world. And at this point, I'd be happy to take some questions.
Questions?
Yeah.
So the cover
So absolutely, some of that is divergent haplotypes that just aren't mapping. Now, at the 10 kb level, we don't really think that that's that big a factor. At a smaller level, absolutely, that is a factor. But at these 10 kb windows, we're merging a number of things together, and so I think we're really just touching the kind of the edge of what we're seeing here. But read depth absolutely is affected by divergence.
So genomic selection is such a powerful application of genomics to reality.
Mm-hmm.
It has been, in animal husbandry, it
Yep.
seems like it's already applied and people are buying meat that's been, improved by genomic selection. How about in plants?
Essentially, it is going on in the two largest seed companies.
Right.
And I think there are, and this is where I think JGI could play a very important role. It is this whole cycle, okay, of being able to genotype fast enough to make decisions and stuff like that is technically hard, okay? And there's lots of minor crops that will essentially not be, and when I mean minor, essentially, other than the top three, okay, may not be able to take advantage of a lot of these opportunities or a lot of other things without help of groups like JGI to get through it. Because it is, technically difficult to pull off selection every 4 months. Right. And it's process hard. It's a systems engineering hard thing to do. Right. And also, I would also want to highlight that these exact same mathematical models we make for predicting breeding can also be used in assisted migration and for conservation. We're going to have to help migrate our natural varieties into the right environments all over the world in the next 50 years. Trees, long-lived species. And genomic selection is the right way to make the accurate predictions to help trees and other things migrate.
Sure. No, I think in some ways we're continually being pressurized by our funders to show an application of genomics. And I think this is really, this is it.
Ed, way back in the corner here.
Yeah, Jeff.
So you mentioned at some point in your talk that the loss of diversity in the worldwide usable maize population compared to the land races was like 2%,
right.
Which is amazing. And then you follow that up by saying that this means there's been lots of low allele frequency, low phenotypic values adaptation. And I'm curious to know whether you think that's really adaptation or actually very high and skilled levels of human intervention instead to select those rare alleles for furtherance and giving rise to capturing most of the natural diversity of the land races in the various used populations.
Right. So I mean, I think our data, and certainly not totally proven, is that most adaptation appears to be based on standing genetic variation that dates back to teosinte. And so I think just because the alleles are out there, they get stacked, that's easier than waiting around for a rare mutation and everything. Now, I don't also think that this, when we talk about the natural standing variation, I really want to highlight that we're looking at this big swath of lines that have also been developed by CIMMYT, okay? And this excellent breeding World Bank and Gates-funded breeding group in Mexico that took land races 40, 50 years ago, mixed them all up, and started deriving inbred lines out of them. So, I'm not saying that they didn't come from the land races 50 years ago. It's just that CIMMYT has already done kind of that hard part of extracting good genetic variation from really diverse materials and making it accessible. So now I do think, and Jeff and I have talked about this a number of times, there are cases, especially I think in disease resistance, where it's being driven by Red Queen and you're always trying to avoid your pathogens, that I think there are going to be very important rare alleles that are going to be in isolated environments that have essentially faced off against various pathogens for a relatively short period of time. And those are likely to be much rarer and more unique than a lot of the common genetic variation. But I think traits like drought tolerance, drought always occurs. It's always been occurring. Waterlogging, lack of nitrogen. These are traits that plants have had to deal with for for hundreds of millions of years, and that's standing variation and adaptations to those have been going on for a long time. Disease, I do agree, and we are working with CIMMYT, and they've got a really interesting design to pull out interesting rare alleles out of their land races. And I think actually the biggest benefits will be in things like disease.
This is related to the last question in a way, and you may have mentioned this number, but I may have missed it. So any two maize plants that you compare have this high amount of genomic variation that you're talking about. Mm-hmm. How much overall phenotypic variation do you see between any two? Is this, I mean, if you measure this sort of globally with lots of phenotypic traits, how does the genomic variation relate to the phenotypic variation between two plants?
So genomic variation does correlate with phenotypic variation, and this is one of those things that people talk about transgressive segregation, so essentially more variation than what the parents have. And I've always thought this was just such a boring question, because I've always been in maize. Every time we cross anything, the offspring are more diverse than the parents are. And the variation's outside. So it's, I mean, to me, this is just what genetic variation is. And maybe that is a perspective in a high diversity species. That's exactly what happens. Nothing is controlled by a single gene or a couple genes that are stacked. And so, I mean, the main reason people don't think maize is very diverse is, the human eye is not very good at differentiating between plants. Biochemically, it's diverse. Every, maize, my colleagues at Max Planck have looked at metabolites. It's amazing how much variation there is in central carbon and nitrogen metabolism in two plants that look to us identically. One may have twice as much malate running around in it, 3 times as much malate. It's there, we just don't see it.
So how many phenotypes can you predict with the data you have right now? And is it the number that is more important or specific phenotypes that you're targeting?
So the most important phenotypes are yield at numerous environments. So there's probably projects going on looking at 100 or so different phenotypes around the world on these, and then lots of sub-phenotypes within those. But the efforts that we don't have any data on are, we're just now getting yield data in a few confined, relatively consistent environments in the U.S. But the ones we'd like to see a lot more work on are nitrogen use efficiency and drought tolerance. And those trials are just kind of right now at a scale that we haven't been able to get the community and the resources to do.
Okay, thank you very much. I'd like to thank both speakers, and we're ready to take a break. Back in 5 minutes.